blood types because blood is often transfused from one individual to another, and this is an equivalent of an organ transplant, and therefore we need to perfectly match the blood of the donor and blood of the recipient. If there is no match, the antigens on the red blood cells in the antibodies in the plasma will react, forming large clusters, just like you see on this image. These red blood cells will be then destroyed. This is known as agglutination reaction. Agglutination reaction in live individual or in vivo are extremely dangerous. The damaged red blood cells will no longer be able to transport oxygen. Moreover, the clusters of those red blood cells will occlude capillaries, and ruptured red blood cells will release large amount of hemoglobin in the bloodstream, potentially causing renal failure. So let's focus on the blood type test. In this test, we will take blood of a patient and combine it with anti-serum containing anti-A antibodies, anti-B antibodies, and anti-RH antibodies. Now let's determine blood type experimentally. We will take a tray with 3 small indentations called wells and apply blood sample of a patient into each of them. We will then add anti-serum with particular type of antibodies into each well. We will start with adding anti-serum A into the first well, and then anti-serum B into the second well, and anti-serum D or RH into the third well. Now we will mix the patient's blood with anti-serum in each well. It is very important that we do not carry the samples from one well to another. That is why I'm using different color sticks for mixing in each well. And of course, I will dispose of them after I'm done. We will let the sample sit for several minutes and then examine it closely for agglutination reaction, in which antibodies in anti-serum react with the red blood cells of a patient's blood, forming aggregations or clusters, like you see in well A and B. When we interpret the results, we will look for agglutination reaction in different wells. If there is no agglutination in a well, that means that there is no antigen present in that particular blood. In this test, you can see no agglutination in the A well. That means red blood cells do not have A antigen. There is agglutination in the B well. You can see individual granules. That's agglutination reaction. So B antigen is present in this blood. And finally, no agglutination in RH well indicates absence of the RH antigen. So no A antigen and B antigen means blood group B, and absence of RH antigen means negative. So this is B negative blood. Let's do one more. I see agglutination in A well, which means A antigen is present. I see agglutination in the B well, which means B antigen is present. So there is AB blood. No agglutination in RH well indicates absence of RH antigen. Therefore, this is RH negative blood. Your conclusion is AB negative. Let's take it a step further. This recipient needs a blood transfusion, but there is no perfect match. Can the blood of this donor be used for the transfusion? Let's first determine the blood groups. Your donor has AB negative group. Your recipient has A negative blood. So this transfusion will result in agglutination because the donor's red blood cells, when they transfused, will be immediately marked as foreign because the recipient's immune system will not recognize B antigen as self. One more. Donor here has A negative blood and recipient here has A positive blood. Yes, we can do this transfusion because the Rh negative red blood cells do not carry any Rh markers, and therefore Rh positive recipient would not recognize those as foreign.